Hi, my name is Rob Holcomb. I'm a PhD candidate in the Institute of Optics working at the Laser Lab with Dr. Jake Bromage as my advisor. Today I'm going to talk about supercontinuum generation in bulk crystals and how we can combine that nonlinearity with the nonlinearities in hollow core fiber to come up with a really dynamic, versatile source. We can also then try to understand the shot-to-shot variations in these pulses and try to search the parameter space for an optimal solution. So we have our system, which starts out with a homemade ytterbium YAG laser. It's a chirp pulse amplification based laser operating at 1030 nanometers. But unfortunately, because of the ytterbium YAG's narrow gain profile, we're limited to pulses only down to about picosecond. And so we need to make those pulses faster in time if we want to reach high peak powers. So what we do is we send it first through a hollow core fiber where we confine the pulse to a gas, in this case argon, where the cell phase modulation inside the argon broadens the spectra and then allows us to compress the pulse temporally to shorter durations. So we found that this intensity-dependent cell phase modulation has a long pulse duration when we're not sending much energy through the throttle. But as we increase the transmission and send more and more energy through the hollow core fiber, we get to shorter pulse durations due to the increase in bandwidth. Next, we send our beam and focus it into a crystal. Inside this crystal, we have carousel focusing, which balances out with plasma defocusing and leads to optical filaments, where we have a self-similar propagation over an extended distance within the crystal. We found that when we increase this throttle, we get increasing hollow core fiber output energy and a decreasing pulse duration, which leads to a decreased energy where we see a threshold for our filamentation and our white light continuum generation. We're then able to study this white light continuum generation to try to understand the process a little bit better. And we do that through a variety of diagnostics. The first one I'll talk to you about today is here. It's a camera that's imaging the side view of our crystal. And so we can take a picture of the crystal where we actually look at the fluorescence from that ionization that took place in our filament. So as the beam ionizes the material, we get some electrons and holes that are separated. And after the pulses pass through, those electrons and holes will recombine, giving off a fluorescence. And so we see in 2D what our filament would look like. And we can extract out that information to see how does the intensity vary as it propagates through the crystal. And it turns out that that changes based on the parameters of our filamentation. In this figure, I'm showing an increasing throttle 2 setting. So we actually increase the amount of energy that we send to the white light generation stage. And at first, at low energy, we don't see any fluorescence in the side view. But above a certain threshold, we start to get these filaments appearing towards the back of the crystal. And it turns out that as we increase the energy, those filaments move earlier into the crystal as the self-focusing distance decreases. We also notice around double this first threshold, we start to see a second filament. And it turns out that there's a lot of energy left over from the first filament that didn't go into a white light generation, and that causes a second peak. They eventually merge together and lead to subsequent refocusing cycles that start to get a little bit noisier and harder to predict as you go on. But in addition to the side view, we're also interested in what's happening to the spectra of our pulse, because we want a spectrally uniform pulse, and we want it to be consistent from one shot to another. And so usually to do that, people use a Cerny-Turner spectrometer. And we actually have one of those in our diagnostic suite, just a traditional ocean optics spectrometer that you can get off the shelf. And one of the benefits of that is that it's really high spectral resolution, but unfortunately it has tight alignment requirements into the input slit. It has a limited dynamic range, and this white light generation occurs over several orders of magnitude. It's usually plotted on a log scale. And finally, we're interested in shot-to-shot dynamics, and the CCD sensors are limited to millisecond timescales. So if we were to increase our rep rate beyond a kilohertz, we wouldn't be able to resolve individual pulses. So instead, we used a photodiode-based spectrometer that we built, which is basically an array of bandpass filters, where each bandpass filters allows us to look at a different spectral region. We can measure those on a photodiode that has a faster time response, so we can resolve individual pulses. After we've extracted this signal, we can look and compare the ocean optics spectrometer to our calibrated homemade photodiode and see that the mean and standard deviation of our homemade spectrometer pulses really agree pretty well with the higher resolution average measurements. We can then use these measurements to look at various trends and try to understand the dynamics of the filamentation better. And so here I'm looking at a bunch of different spectral channels and plotting them as a function of input energy that we recorded on our 8th photodiode. And so we see in the insets of this figure the fluorescent side view. And at first there's no fluorescence, and that kind of agrees with our output where we're not seeing any broadband filamentation or any continuum. But eventually we strike some threshold where we got the self-focusing and catastrophic collapse into a filament, and we see an extended region of relatively stable filamentation. In the side view, we see a single filament before reaching additional thresholds where our noise increases. We see a second filament, which then moves further and merges with the first and generates a third filament. Eventually, these get really noisy. Our signal gets to a point where you might not want to use this in a larger system, and the side view reflects that. There's a lot more going on that's harder to predict. The other, besides the first, second throttles that control the pulse duration and the total energy, is a linear translation stage that our crystal sits on. And that controls where is that focal position relative to the front of the crystal. As we focus deeper into the crystal, we see different trends in our 7 spectral channels. If we focus before the crystal, we really don't see much spectral broadening. About 1 millimeter before the crystal, we start to see a little bit of broadening coming from the red edge, coming from that main driving wavelength, but the noise is relatively high relative to the pulse signal. As we focus deeper into the crystal, we can start to observe more of what we'd think of from a standard filament. And we see an extended region where the noise in some of our spectral channels is relatively low. If we focus deeper into the crystal, eventually our side view shows a cutoff where the filament actually reaches the end of the crystal before all of the white light generation has occurred. And so that kind of causes our beam to cut off. Other things that we see is a little bit of a spectral tilt. So if we focus earlier in the crystal, we see more energy in these spectral ranges around 800 nanometers and less in the blue edge. But if we focus deeper in the crystal, we see more energy in the blue edge than we would the red. And so between these 3 parameters, we have a lot of knobs to turn that could help us to get desired properties out of our filamentation and end up getting a stable seed that's spectrally uniform and well understood for a wide variety of applications. Thank you.